So, in this model we will discuss the various antibacterial assays. Let us recall that in one of the earlier modules we have discussed the bacteria structure and characteristics. So, just to quickly refresh our mind, there are two type of bacteria, gram positive and gram negative bacteria, and out of that gram positive, particularly Staphylococcus or Staphylococcus aureus, S aureus and S epidermidis, they are like much they are known for the pathogenicity, or they are like more pathogenic bacteria. So, to cause any bactericidal effect or to kill these bacteria that which are pathogenic in nature, that is extremely difficult. So, there are two ways that one can deal this bacteria, this antibacterial and to induce antibacterial property. One is that there is something called antibacterial material. So, just to show you the, or just to illustrate that how to develop this kind of material, is it like hydroxyapatite is widely investigated because it is the inorganic constituent of the human natural bone, but hydroxyapatite as such does not have any antibacterial property. Like if you grow E. coli bacteria, which is very well known gram negative bacteria, then E. coli bacteria can happily grow on hydroxyapatite as such. So, what people do, people use certain antibacterial phase like silver for example. So, they add silver to hydroxyapatite to induce that antibacterial property, like the presence of silver will facilitate bactericidal property. Another material that people use like zinc oxide. So, people use that, add zinc oxide, the researchers kind of develop that zinc oxide reinforced hydroxyapatite or zinc oxide added hydroxyapatite. Now, the role of silver zinc oxide to induce bactericidal property. So, in this context there are certain terminology which is important to induce bactericidal property. So, there is two terminology, one is bactericidal versus bacteriostatic. Bactericidal means that the materials with the capability or certain antibiotics with the capability of killing bacteria in growth medium. Bacteriostatic means the materials with the capability of inactivating the growth of the bacteria. That means, if you grow the bacteria or if you want to grow the bacteria on these particular materials, those bacteria may not be killed, but at the same time those bacteria may not undergo binary fission process. So, that 1 to 2, 2 to 4 like that their growth would be inhibited. So, in other words, let me summarize this. So, it is growth inhibited in case of bacteriostatic property or inactivation of bacterial growth and bactericidal means killing of bacteria or bacterial death. Okay. So, these two things should be clear. So, let me summarize these two terms. what is I have mentioned bactericidal means bacterial death or killing of bacteria. Bacteriostatic means growth inhibited or inactivation of bacterial growth. That does not necessarily means if you have a bacteriostatic property it will kill all the bacteria. So, one of the examples of that bactericidal property that or materials exhibiting bactericidal property, hydroxide silver, hydroxide zinc oxide. So, these kind of materials. Now, other things that silver or zinc oxide, although they have a antibacterial property, now this addition to be tailored in the context that so, what I said, for example, hydroxyapatite you add this material x% silver or x% zinc oxide. So, this x should be such that it will cause bactericidal property without compromising or without inhibiting cell growth or functionality. Here growth and functionality in the context of eukaryotic cells like truly nucleated cells. So, that means this addition of silver or zinc oxide to induce bactericidal property should not in any way inhibit the cell growth or functionality behavior on that particular substrate. So, these things needs to be very carefully tailored or very carefully kept in mind while developing these kind of antibacterial materials. Now, once you develop these antibacterial materials, next thing that one should understand that how to quantify or how to assess both qualitatively and quantitatively this bactericidal property. Now, for that one is that strain type dependent bactericidal property needs to be assessed. What it means, for example, gram positive, if you take Staphylococcus species. Now, Staphylococcus they have number of strains like Staphylococcus aureus, Staphylococcus epidermidis, and this is the gram positive. In the gram negative mostly people use Escherichia coli. This is the gram negative bacteria. Now, all these bacteria, so if you use just only one type of bacteria, it is not sufficient to establish bactericidal property, but instead one has to do more cell types or more strains like S. aureus after the S. epidermidis, so that you have the bactericidal property not only one bacterial strains, but also multiple bacterial strains. Not only one bacterial type, let us say gram positive, but also gram negative as well. So, these things must be mentioned, these things must be carefully considered, so that this cell type or strain type dependent property can be established. So, one single material may have good bactericidal property against E. coli, but that does not mean the same material will have equally good bactericidal property against S. aureus. So, it may not be able to kill all the S. aureus bacteria when this S. aureus bacteria will grow on that material substrate. Now, to confirm this, typically in biology it is very common to use not only one cell type or only one strain type, but multiple of those to confirm certain evidence or to confirm certain mechanism whatsoever it is. Now, just to refresh your mind on that biofilm formation and mechanistic description of the same. So, initial stage is that 1 or 2 bacteria will come and adhere in this material substrate. Now, if you remember, the bacteria they grow much faster than eukaryotic cells. Like if the typical cell division type cell division time of eukaryotic cell or human cell is 11 to 12 hours, bacteria the doubling time of bacteria is 30 to 40 minutes, most of these bacterial strains. So, so therefore, the moment it adheres or it gets attached to a material substrate, then these it undergoes binary fission from 1 to 2 bacteria and so on. And there are 2 things that has been mentioned here, that is that in one case it is shown that if you put some surface coating on the material or substrate coating, and another case uncoated substrate. Now, this colonization would be there, but this colonization would be more specific to the part where certain specific coating is given to the bacterial to the biomaterial substrate. Now, once this cell monolayer forms, then there is polysaccharide secretion, and this polysaccharide would act like a glue, and then that will essentially help in the formation of some bacteria clusters. Now, these clusters will grow and then it will form biofilm, and this biofilm, then once it matures biofilm, then any more bacteria which will come from the growth medium, that bacteria cannot get attached to this biofilm, and as a result those would be detached. And this actually tells you that bacterial biofilm grows to a kind of a critical size, and therefore, this biofilm cannot grow any further. So, all these things needs to be kept in mind while assessing the or while using different antibacterial assays. So, this is the typical protocol for the bacteria culture, like you know that you start with this sample preparation, then you grow the bacteria, and then before that you have to do sterilization, then you grow the bacteria in an incubator. This incubator is 37 degrees Celsius. There is no requirement for the CO2 flow unless eukaryotic cell culture, because bacteria can grow in normal ambient environment, and then after that you fix the cells by PBS, phosphate buffer saline. After that you can do either critical point dryer or you can do HMDS, hexamethyl disilane HMDS chemical. Then you fix it, then you can see either ACM or live date staining using fluorescence microscope. Now, these are different assays that is important, and these assays are MIC, MBC. MIC stands for minimum inhibitory concentration, and in a planktonic culture, like when the bacteria is kind of suspended in a Appendorf tube, not actually on the growing on the substrate. Now, this is there is zone of inhibition ZOI, and there is CFU, that is colony forming unit. So, there is 4 things MIC, MBC, ZOI and CFU. These 4 things, these 4 aspects need to be assessed qualitatively and quantitatively. So, let us see that what is the meaning of this kind of what is the meaning of these terms. Let us define that. So, minimum inhibitory concentration means lowest concentration of any antibacterial, any antimicrobial. Now, here in the context of the bulk materials, you can say that HA X% silver or zinc oxide. Now, X to be optimized, that is what is the minimum percentage of the A or minimum value of X which will give you that inhibitory concentration. Now, if it is, let us say take the example of the gold nanoparticle. So, AUNP, what is the dosage of this gold nanoparticle in solution which will cause this MIC, that needs to be identified, which inhibits the visible growth of a microorganism. It is characterized by the absence of any exponential phase in the bacterial growth curve. If you remember, the bacterial growth curve goes like this, it goes to stationary phase, and then that needs to be so essentially MIC is characterized by the absence of the exponential phase in this one. Second one is the minimum bactericidal concentration, MBC. that is the lowest concentration of an antimicrobial agent that kills a bacterium. So, it is an extension of a MIC test, but wherein the both the cultures are contain greater than MIC doses which are subcultured on the solid agar plates. So, what it means that, so you do that MIC test, you extend this test further with these doses of greater than MIC and to see that what is the minimum bactericidal concentration. Third one is the zone of inhibition assay, that efficacy of any antibacterial in preventing bacterial growth, and this is the zone of inhibition, and one has to see that where is the no growth or no growth of the bacteria around the antibiotic, and this is the referred to as zone of inhibition. As you see that most of these definitions are essentially centered around the qualitative or quantitative effect of an antibiotic, which is been traditionally used as a drug or antibacterial drug. Now, in the context of the biomaterials, the drug dosage has to be converted or has to be understood with respect to the antibacterial phase content in a particular biomaterial substrate. So, let me repeat what I said. I said, let us say there is the antibiotic drug A, you want to understand that what is the dosage X or dosage Y, which is like minimum inhibitory concentration or which has a ZO, which can be assessed for the ZOI or for the MBC. Now, in the context of the microbial antibacterial material, let us say hydroxyapatite, and I have given multiple times this example X% silver composites. So, now you are interested, instead of antibiotic, you are interested what is the value of X% silver, which will give you the similar description for the MIC or MBC or ZOI. So, antibiotic here is equivalent to the antibacterial phase or antibacterial material or antibacterial compound like silver or zinc oxide, which is added to another material to induce the bactericidal property. I hope I have explained you sufficiently so that you understand that this definition. Then colony forming unit, that is the number of bacteria counted by the volume of culture plate. Volume of culture is plated multiplied by dilution factor. This is also one of the quantitative aspect of this antibacterial property evaluation. Okay. Now, while analyzing that why a bacteria is not able to grow or why a bacteria is killed in growth medium, certain assays typically is used to see the effect on the bacterial membrane itself, and this is called inner membrane permeabilization, and this one of the assay that is done, it is called WNPG assay. So, in the WNPG assay, as I have described, as it has been described in this slide, so WNPG is a particular biochemical, or this WNPG when it is added to this one into the bacterial growth medium. So, you have a beta galactosidase, and this WNP is WNP react with that one, and then you WNPG reacts with this beta galactosidase, and then you get WNP which can be detected at 420 nanometer wavelength, and then one of the product is galactose. The second one is that outer membrane permeabilization, that also that is also used to quantify whether there is any instability in the membrane integrity, and that is uptake of a certain fluorescent. This is NPN is one of the fluorescent by membrane permeabilized cells. That means, if the membrane becomes more permeable or if the bacterial membrane their integrity is disturbed or integrity is lost, then what happens? It will undergo it will undergo death phase. So, so this comes to the next description of the membrane integrity assay like LDH assay, which I have described earlier in the context of the cell viability, cell death. And if you recall, there I have categorically mentioned that for the cell viability assay or for the cell cell death quantification, you need to use these assays in a complementary manner. For example, MTT assay will tell you that how much fraction of the cells are viable, LDH assays will tell you how much cells are dead. So, these MTT and LDH assay needs to be done in a complementary manner to quantify what is the fraction of the cells which are growing on a material substrate is alive, what is the fraction of the cells on a material substrate is dead. Now, LDH is an enzyme as been mentioned, and then LDH, when it is a bacteria is damaged or the membrane integrity is lost or disturbed, then what will happen, NDH will come from the cytosol or cytoplasm to outside the culture medium, and then the LDH can be detected at an absorbance of 260 nanometer. And all these biochemical reagents or these any enzymes, they are essentially detected at different wavelength that has been measured, that has been mentioned earlier also. Okay. Like in eukaryotic cells, one can also do precise cell level cell fate determination for bacteria also, and in case like eukaryotic cells, one can use fluorescent activated cell sorter, which I have described significantly when I discussed the cell fate processes. So, you have two things there, one is side scatter, one is forward scatter. Side scatter is SSC, forward scatter is FSC. Now, what is the applications of the flow cytometry? That is, one is the cell apoptosis or necrosis in the context of eukaryotic cells. In case of bacteria, it will be simply cell death or cell, what is the cell viable or so what is the cell death. Oxidative stress, ROS generation, that can quantify. Cell cycle analysis, which is very appropriate for the eukaryotic cells, cell counting, cell number, and cell sorting from mixed cultures. Now, out of that, in the context of bactericidal property, often the presence of ROS is attribute or ROS is shown as a reason for the bacterial death. So, therefore, oxidative stress needs to be quantified, and this oxidative stress, as per definition, it is present either as superoxide or hydrogen peroxide or hydroxyl ion. These comprise that reactive oxygen species, and this nitric oxide is an example of the reactive nitrogen species. So, you have ROS, reactive oxygen species, you have RNS, that is reactive nitrogen species. So, reactive nitrogen species is this nitric oxide, and ROS is essentially H2O2 or O2 superoxide and so on. So, this is the fundamental equation reaction, that is Haber-Bosch reaction, that is essentially shows that how ROS is generated in the intracellular region of a bacteria, and then it finally gives rise to hydroxyl ion, reactive hydroxyl ion, which is one of the constituent of the reactive oxygen species. So, once you get this reactive oxygen species, then the next stage is that you have to quantify this reactive oxygen species, and then you can do that using that fluorescent activated cell sorter. And this fluorescent activated cell sorter analysis, this fluorescent activated cell sorter, what has been shown here in the case of the live dead assay, you use the propidium iodide, and in case of bacteria you use the different reagent is called thiazole orange A. So, this use of these two reagents will essentially stain the cells differently. In one case you have that unstained debris, and green dots are like live cells. If you recall, the fundamental principles in these facts is that each dot in the facts plot essentially comes from one individual cell. Okay. So, therefore, the multiple of these dots which are stained green, that means this means so many numbers is the live cells. Okay. That this particular coordinate you have this particular quadrant you have these injured cells, and this is kind of membrane compromised cells. So, from this particular example here, if so many fraction of these white green dots are there, that means close to 90% or more cells are essentially live cells, live bacteria. Rest of them are either injured or unstained debris or membrane compromised cells. Now, coming to ROS quantification using fluorescent activated cell sorter, this is also alternatively known as DCFDA assay. So, you use this DCFDA and as a reagent, biochemical reagent, and then what happens if this particular reaction is taking place at the intercellular region whose end product is one of the constituent of the reactive oxygen species, then this constituent, if it is there, then what will give? It will give some green fluorescence, and this is your controlled cell population, and this is your treated cell population, which is shifted towards the right hand side here in the DCFDA, and from there you can see that what is the fraction of the cells where the ROS quantification is significant enough, and then you can quantify this, what is ROS that is generated. So, to go back to this all this summarize what I have essentially taught you so far. You have to quantify what is MIC, MBC, that minimum inhibitory concentration, minimum bactericidal contents concentration, zone of inhibition, colony forming unit, which will give you the quantitative aspect of the antibacterial ability of any material, and these parameters were defined in the context of the use of antibiotic, but now it has been defined in the context of antibacterial material. Now, once you do these things, the reasons for any antibacterial effect or bactericidal effect is to be analyzed with respect to some additional assays like inner membrane permeabilization, outer membrane permeabilization, or LDH assay, which will give you the membrane integrity assay. So, the rationale is that first you quantify whether any material has sufficient antibacterial property, then you try to propose certain reasons to explain the origin of this antibacterial property of that particular material. Now, to support your rationale or support your logic, you have to do some additional assays to confirm both qualitatively and quantitatively that it is indeed that it is because of the membrane permeabilization or membrane integrity, disintegrity, that this bacteria is being killed in contact with the materials, or the bacteria is not able to grow once it is coming in contact with these materials. And also more biological origin may be, or biophysical origin can be reactive oxygen species, and then you need to be quantified that ROS using FACS. Now, up to this part it is done. So, then you confirm the antibacterial. Next stage is that if a bacteria does, if a material has some indication that it has very strong antibacterial effect, but still detectable number of bacteria has the possibility to grow when this bacteria cultured for extended time period in the growth medium, and they can potentially form the biofilm. Now, this aspect of the biofilm formation I have mentioned very briefly in this module, but I have mentioned more extensively in an earlier module where I have covered bacterial growth and bacterial bacteria structure and characteristics. Now, this is that is this is the scanning electron images. This is the ACM image of the biofilm. As you can see that these are like spherical shaped Staphylococcus species, either S aureus or S epidermidis, and there is some ECM like features which is not very dense, but it is more like a porous kind of surface layer which is forming, and which kind of connects that these spherical species, and this is like polysaccharide rich biofilm which is formed on this material substrate. Now, this biofilm also one has to quantify this by different assays, and several assays is been mentioned here. One is called Resazurin dye reduction, one is called crystal violet staining, third one is called live date biofilm imaging. So, you can do confocal microscopy to do that, and one is called biofilm thickness, that what is the typical thickness of the biofilm, that is to quantify. Now, once you do all these assays, the last stage of characterization can be, which may be little more difficult, is called transmission electron microscopy observation of the bacterial cells. Now, this transmission electron microscopy, which itself is a one of the higher level microscopy techniques to see that ultrastructural features of the bacteria, which you cannot probe using scanning electron microscope or fluorescence microscope, where the magnification, allowable magnification is much less than transmission electron microscope. The first hurdle in the using transmission electron microscope for cells and bacteria is to appropriately prepare the sample. The sample preparation is the most difficult stage in TM investigation, particularly for the biological samples like cells or tissue sections. So, there is a technique called ultramicrotome. So, this ultramicrotome, if you do this, so there you can essentially measure very thin sections. So, what you do, you first grow the cells in certain growth medium, and then you allow the bacteria to grow, and then one of the technique in the TM sample preparation that one has to be careful while using this biological sample, one is to use osmium tetroxide OSO4 or glutaraldehyde. Now, osmium tetroxide, the use of osmium tetroxide will give sufficient contrast in the electron micrograph of the cells or bacterial sample. In the normal material science samples, one never uses this kind of additional supplement like osmium tetroxide and so on while preparing the TM sample. So, you need to know how to prepare these TM samples before you can go even to the transmission electron microscope to see the ultrastructural features. Now, to come back to the discussion again in this slide, what I am showing that, so first you have to grow the cells, then you have to fix the cells on certain substrate. After you fix it, then you do dehydration and all these things, and then cells embedded in white resin or within gelatin capsule, then you can. So, essentially you should use certain substrate to hold or to for the attach the bacteria or cells or tissue before you can very thin, you can prepare very thin sections, and for which you have to use ultramicrotome. So, ultramicrotome once you use, you get very thin sample, you can use glass or diamond knife, then picking up these section specimens on the grid, and once you grid it, then again you do some additional treatment called, you can do staining using uranyl acetate or lead acetate. Only after doing all these things, if you go to TM and you put biological TM in the sample chamber, then this is the example of a Staphylococcus aureus species, and it is treated with gold nanoparticle, and after it is treated with gold nanoparticle, you can see certain features like whether these gold nanoparticles are internalized inside the cytoplasm of this bacteria, and if it is internalized, you can then correlate that observation with the reactive oxygen species that you might have quantified before using FACS. So, I am trying to give you some indication that how these TM observations can be helpful to correlate with some of the other features which you have quantified as part of your total investigation. Now, just a few last things, that what are the information that you can get or you can extract from transmitter microscopy. One is the cell wall thickness, what is the cell wall thickness, and there you can use certain control sample. So, in the context of gold nanoparticle treatment, you can grow bacterial population without gold nanoparticle, with gold nanoparticle. With gold nanoparticle will give you the normal cell wall thickness or equilibrium cell wall thickness. In the gold nanoparticle treated bacterial morphology, you can see whether there is any thickness reduction because of the membrane permeabilization or because of the disintegrity of the cell membrane. What is the dividing cell frequency, and is there any preferential localization of the nanoparticle or drug within the bacterial, for example, specific protein or DNA, whether the gold nanoparticle gets attached to this DNA or not. Those kind of things you can very well describe both qualitatively and quantitatively using transmission electron microscopy of the bacterial cell specimen. Thank you.